year or even this semester. Some of these, though.
Prokaryote. We should start at the beginning.
Start at the beginning?
And then get through up until whenever we end.
Like get through the flowers.
So the whiteboards are fine, but the problem with the whiteboard is that you don't end up with a picture, right? So that's why I had the paper. Honestly, if you want to use the whiteboards, they're right here. Go ahead. Not an issue, and I don't want to tell you how best to study, but yeah, feel free.
There goes
it. Also,
Hey guys.
Okay.
You do
Yes.
So the semi-conservative model is where you are drawing all the enzymes replicating the DNA, or is that not the same thing?
It's more like you are drawing a helix that breaks apart and has a new strand of DNA, and what you're labeling is the 5s and the 3s and the 3s and the 5s.
So
It's very straightforward.
what is the one with the enzymes doing stuff?
You don't need to draw that one.
Oh, where's the header?
And here's the other, brain research suggests that when you draw things, you learn them better. A good number of the diagrams you've drawn in your learning targets are not actually testable, but they help you to like associate or understand a concept. To the best of my knowledge, these are the testable ones. And again, if you've looked over any of the paper twos, or even the one I put up on Google Classroom, diagrams are not a huge part of the test generally. They'll probably ask you to draw 1, maybe 2, who knows. But again, the reason we're doing this is because I really, really believe that in drawing them, we will be reviewing a lot of the concepts related to them. And that's what we want to do. So let's go ahead and start with the prokaryote. Everybody ready? Let's do this. Prokaryote and its parts. Draw a Twinkie. I'm aiming for closed lines and neat labels and lines that don't cross each other. Of course, we don't want to use arrows to label things. And what we just drew is the cell wall. We'll get to that later. The cell membrane is just inside. And then inside of that, what we have is a looped DNA region called a plasmid. You don't need to go crazy with the looping. You could even just do a real big squiggle, is what I usually did in class. But it's a loop of DNA. We've got the cytoplasm here, implied. There are 70 S ribosomes in the cytoplasm.
That's enough stuff.
You don't need to go nuts with the ribosomes, just have a few of them. Some prokaryotes have flagella, so we will probably put flagella on there. 3 is generally a good number. And the last thing to put on is these little structures called pili. And again, with diagrams like these, they are not going to go and say, oh, that person only drew 23 pili. They were supposed to draw 24 at a minimum. The guidelines are represent the structures, label them accurately, and go from there. So, label them in the same order we did them. This is the cell wall. And just for the record, I'm not going to put the annotations on the board unless you folks really want me to. I'm just going to label them, and it'll be sort of up to you to maybe go back and find in your notes what all of these things do. I will tell you what they do right now, though. This one provides protection and support, right? It gives a little bit of structure, a little bit of shape. It is not made of the same stuff as the plant cell wall. Cell membrane does the same thing. Regardless of the cell, it controls the entry and exit of substances. This closed loop of DNA region, you could call it DNA. You could also call it a plasmid. It contains the genetic material for the cell. I would recommend that you label a couple of these just so they know that you're labeling the dots. 70 S ribosomes. The big difference is that they are smaller. The flagellum, there's an A in there, sorry. Flagellum. And flagellum are pretty exclusively for movement, that's all they do. And the pili, and there's a little bit of inconsistency on if you spell pili with 2 L's or 1. I'm going to go with 1 for right now, because I feel like it. And the pili do a couple of things. They assist with attachment to surfaces, and they also assist in DNA transfer. Cool. Any questions on a prokaryote? Yes, Amelia?
Not on the prokaryote in general, but like with labeling techniques. So if like you are doing 17S ribosomes and you're like, oh crap, I just drew one and I already have a line, could you like in theory make it more of like a tree type thing that goes to one baseline instead of 2 lines meeting up at one point?
You mean something like this?
Yeah. Would that be considered bad form?
I don't think that's bad form.
Okay.
Really the reasons that I have these rules and these conventions for drawing is because if you don't have them, you guys have no idea how bad the diagrams get. They get really awful. Just strange lines going places and arrows that you don't know what they're pointing to and things crisscrossing and
The crisscrossing.
yeah, it's nothing we want to worry about. Okay. Cool.
No.
We ready to move on? Try another one?
Yes.
Let's do it.
That's
All
a
right.
prokaryote, right? The one inside the.
Oh yeah, I'm sorry. The best name for it would have been the nucleoid.
Good afternoon, everybody. Just a reminder, there is an insurance meeting for all employees to hear about the open enrollment sign-up coming up. Any questions you might have about insurance, it is in the media center, and we'll start promptly at 3:30. So head on down to hear about insurance options. Thanks.
What'd you
You
say?
share because you care.
That's true. Mr. Larson, everybody. She's got some wisdom.
Just as fun right now as it always was.
Fair enough.
Alright, how long are you going?
Probably midnight. We'll see.
Good luck.
Yeah. Lock the door. I'm just kidding. I'm kidding.
Is he?
He's actually locked. All right.
No, studies.
Oh, Larson.
Yeah.
He's saying we're studying.
Okay. Let's do a plant or an animal cell. Not both.
Plant, yeah.
Plant it is.
Hello? Plant.
And then can we just like briefly discuss the differences?
Yes.
Okay.
Well, I'll make a little table off on the side of the big differences.
Okay, that works.
All right. So the main thing you're going to want to do if you're doing a plant cell is you're going to want to make sure that it is boxy. A hexagon is a great way to do this. Which already is one of the main differences between plant and animal cells, is that animal cells are usually rounded, plant cells are boxy. Doesn't matter if you do it on the inside or the outside, I'll just do outside, but you're going to want to give this one a thick cell wall. Another one of the main differences between plant and animal cells. Now the cell wall should have thickness. So this is not the cell wall, and this is the membrane. This is the cell wall, so it's going to be a little weird. You're going to put another line on the inside, and that's going to be the membrane, just inside. All cells, for the record, have a cell membrane. Okay. Animal, plant, boxy versus no, cell wall versus no. Okay. On the inside, the big thing that you're going to want to label, and you can really go crazy labeling this. I would say, in general with these diagrams, label everything that you can. I can't think of an instance where having labels that aren't on the mark scheme would count against you. It's only how much you have and how much they're looking for. So let's go ahead and give this puppy a nucleus. A nucleus is one of the structures in the cell that has 2 membranes. The other structures in the cell that have two membranes are mitochondria and chloroplasts, both of which will be in this plant cell. The nucleus also has these nuclear pores. Nuclear pores allow mRNA to enter and leave. On the inside, I would just go ahead and give it some, I hesitate, and you could do the squiggle. That would be fine. Squiggle's fine here. I was going to say give it some chromosomes, but chromosomes aren't necessarily the most appropriate thing unless they're in mitosis. Chromosomes only appear when mitosis is happening. Outside of the nucleus, you'll have two endoplasmic reticulums. One of them is the rough endoplasmic reticulum with ribosomes on it. The other one is a smooth endoplasmic reticulum. You can draw them the same way, just don't put ribosomes on it. Now, animal cells have centrioles. Centrioles we usually drew as two little cans, and it is from the centrioles that the cytoskeleton comes, and this is what grabs things in mitosis, the spindle microtubules. Plants do not have centrioles.
Do they have lysosomes?
They do. So whereas we would put centrioles in our diagram if this was an animal cell, we do not have them in our plant cell. We do have a cytoskeleton though, so they're just going to kind of come out of nowhere. And you don't need to make it super expansive or extensive. We'll give this guy a mitochondria, and the best way to do a mitochondria is an outer membrane and then a foldy inner membrane, but it should still be a circle, just a foldy circle. Chloroplast, same basic idea, about the same size, about the same shape, but on the inside of a chloroplast there isn't a second folded membrane as much as there are, it basically looks like a stack of pennies. Plant cells also have lysosomes. Or, you know what? This is the part where I get fuzzy. Plant cells actually don't have lysosomes. That's one of the differences. Thank you, Amelia, for making me think of that.
And animals don't have chloroplasts.
Animal cells do not have chloroplasts. Thank you.
How would you draw a lysosome?
A circle.
It was just a circle.
Just a circle.
Do plant cells have ribosomes in the
They do.
cytoplasm?
And I'll just label this one right now. Starch granule.
Animal cells don't
ADS ribosomes in the cytoplasm.
Animal cells also don't have starch granules, correct?
Animal cells would not have starch granules. Starch is a polymer that is specific to plants. Your version of starch is called glycogen, and it's in your liver.
How did you draw the thing that's inside the chloroplast? Is that just like?
It's just a little stack of like pennies or pancakes or
okay.
something like that.
Do we have to draw these little holes in the wall?
Oh, this is the mitochondria.
In the vacuum.
Okay, and there's, I think there are two more things to add.
I don't have enough room for a vacuum.
A large central vacuole, and then a series of folded membranes that we would call the Golgi apparatus.
Weren't those the pancake ones?
They kind of also resemble pancakes. These are just sort of a different kind of cellular pancake.
Does the Golgi have to be like right there?
It's good to have them near, because this is sort of a pathway for exocytosis right here. Yeah, Joy?
No, I was going to ask if it needs to be just like that.
It doesn't need to be exactly like it. You could probably put the Golgi apparatus anywhere, and as long as you label it correctly, I think you'll be fine.
Oh.
So, yeah, Aaron?
So cholesterol is not how animal source starts, that's something else.
Cholesterol is a saccharide in animals. It stabilizes the membrane, but it's not a polymer. So, I'm going to go ahead and label these real quick for us. Cell wall provides support and protection and structure, rigidity to the cell. The cell membrane allows the entrance and exit of substances, once again. I'm just going to go nuts here with the nuclear labeling. This is the nuclear membrane, which allows things in and out of the cell. I'm also going to label a nuclear pore, the means by which substances would travel into and out of the cell. Here we have the DNA in chromosomes that are not pictured. It's worth noting that chromosomes are pretty unique to eukaryotic cells. And if you find an extra dark labeled region, you could label that the nucleolus. And that's all the stuff I could think of to label on the nucleus. It's a good number of stuff.
Or just the nucleus itself.
And, thank you. And the nucleus itself. Okay. Let's go nuts here. Yes, Manir?
Like on the actual exam, can we use a pencil for the drawing or use pen?
I think you have to use a pen. You have to use a pen.
So
But Ms. Baker is correct when she says that if you make a mistake, you can just cross it out and do another one, and they're not going to label the one you or they're not going to grade the one you crossed out. And you also don't need to go crazy crossing stuff out. Just make it clear that's not the one you want. You don't need to like scribble it until you're like it never existed. Like you don't need to spend that much time on it. All right, let's keep going here. This is the rough endoplasmic reticulum. It makes proteins that are destined for exocytosis. Exocytosis in a vesicle that goes to the Golgi apparatus and then leaves the cell. The smooth endoplasmic reticulum generally contains enzymes for detoxification. The Golgi apparatus is sort of like the FedEx of the cell. I'm writing down its annotation right now. I was going to say it modifies, packages, and ships proteins, but I'm going to write Golgi apparatus. It modifies, it packages, and it ships proteins. And though I wouldn't put this on your diagram, this is a pretty good through line, especially in animal cells. DNA makes RNA, RNA hits a ribosome, makes a protein, protein leaves in a vesicle, is finished in the Golgi apparatus, hits the membrane, the bubble fuses with the membrane, and it releases the proteins out of the cell in exocytosis. This here is the cytoskeleton. Vacuole. Cytoskeleton, transport of objects around the cell, a little bit of movement, though not very much in plant cells, but they can change the shape of animal cells, the cytoskeleton.
There's no vacuole in animal cells.
80S ribosomes.
Yeah.
Whew. Chloroplast. Mitochondria. Starch granule. I think I actually forgot to label, but it wouldn't be a bad one to label. This wasn't on the prokaryote, but the cytoplasm. Anything else? No, I think that's good. Chloroplast produces glucose in photosynthesis. Mitochondria produces ATP in cell respiration. And if we want to kind of keep going with our list over here, the plant cell has a vacuole. Animal cells do not. If I remember correctly, there are 3 things that the animal cell has.
Starch granules.
Flagellum.
Flagellum, that's it. It can have a flagellum. All right. Any questions on that one? Ooh, no.
Do we get points taken off? Because I remember vaguely when we did this unit like last year. Do you get points taken off if you write RER and SER instead of the full
Point for that on
name?
my first.
Do not abbreviate anything unless you give it the full name first and then reference that later.
Okay.
So if you were writing an essay, you might say, the rough endoplasmic reticulum, in parentheses, RER, and then you can call it RER for the rest of that essay. Yep. Don't abbreviate anything, if you can avoid it.
Is it okay to like abbreviate those not proteins, the hormones like in the pregnancy stuff that we did last unit?
I would, again, same rule. I would say, call it by name once, and then you can abbreviate it LH, FSH, etc. One other thing that's worth bringing up here is if this was to do a mitosis event, if we were to do cell division, you might get a question that's like, oh, what's the difference between cell division in plants and animals? There's one big difference.
Cell wall.
Cell wall. You got to build a cell wall in the middle of your cell if you're doing plant cell division. Mr. Andrew? There's a pizza delivery. Thank you very much.
Kyle, what?
Oh my god! I wasn't expecting.
I think I need to sign for it, so I'll go down. I'll be right back.
I can tell.